Hello students, good morning. Today we are continuing the same topic. Yesterday we have discussed about lot in carbohydrates, and also we have discussed details of monosaccharides and some of the properties of the monosaccharide, and these some of the properties we have discussed related to the physical properties, and then we also looked or started with classification of a monosaccharide. Classification of a monosaccharide based on their number of carbon atoms present in the monosaccharide, as well as we are looking into the other kind of a classification of this monosaccharide, so as to we will be making a clear idea about the monosaccharide. So yesterday, as in the classification of a monosaccharide, we have discussed in detail that monosaccharides are classified based on the number of carbon atoms, such as if monosaccharide contains 3 number of carbons, then it is a trisaccharide, triose. If it is containing 4 number of carbon atoms, then it is a tetrose. 5 number of carbon if contains, then it is called as a pentose. 6 number of carbon, hexose. 7 number of carbon, heptoses. And we also discussed in detail that what are the different kinds of the examples and what is the general formula of those different kinds of the monosaccharides. So today we are going to discuss the next important classification of this monosaccharide. So these sugars are also can be classified based on the type of a functional group present in their chemical structure. So second type of a classification of this monosaccharide is based on type of functional group. Type of functional group. So usually there are 2 different kinds of functional group we find in the monosaccharide. One, the aldehyde functional group, aldehyde, and another ketone as a functional group. So these 2 different kinds of the functional group we do usually find in the monosaccharide. And so because of the presence of any of the or either of the functional group present in the monosaccharide, monosaccharides can be classified into the 2 different groups. So based on the functional group, these monosaccharide can be containing the aldehyde, and therefore we can call it as a aldoses. As yesterday we have looked into the, or we also seen that type of how we can name the sugar or monosugar. If suffix of OSE is there, then you can consider it as a sugar, and therefore aldehyde name is modified by putting a suffix, that is a OSE suffix, and therefore the name becomes a aldose. So such kind of the sugar which contain aldehyde are the aldoses. So what are the different kinds of the aldoses? So based on the number of carbon atoms as well, we can think in the details of the aldoses. So for example, if we are thinking of a triose, what kind of a triose is a aldose? Triose, in the triose, if aldehyde is a functional group, in triose sugar, if the aldehyde is a functional group, or you can say CHO group, so this CHO group, if contained in the triose, then that triose, very simple triose yesterday, also we have taken an example, that is a glyceraldehyde. Glyceraldehyde. So here aldehyde is a functional group, therefore this 3 carbon sugar is a aldotriose. You can say aldotriose, and so this glyceraldehyde can be called as a aldotriose. You can remember aldotriose. So another 4 carbon sugar, tetrose, erythrose, or you can, there is a no, or there is a no such kind of examples you need to remember, but you can have many different kinds of the examples of such kind of trioses, as well as tetroses as well. Tetroses, aldotetroses, aldotetroses, pentoses. If pentose contain a aldose group, those can be called as a aldopentoses. And if it contains a 6 carbon number, those are the hexoses, and hexoses containing the aldehyde group, those are called as a aldohexoses. Likewise, you can make the important sugars, that is two different kinds of the important sugars are there, that is a pentoses and the hexoses, or you can say aldopentoses and aldohexoses. Then there can be some many different kinds of the examples of the aldopentoses and the aldohexoses. So aldopentoses, if it is a 5 carbon sugar, there can be the different kinds of the examples. So there is a one important example we have mentioned yesterday also about pentose, and that is a ribose. I will first write examples of this pentose, aldose, and that is a ribose. Ribose is a aldopentose. And when we look into the hexose, then these are called as aldohexose, and a very common example of aldohexose is glucose. So these are the many different kinds of the aspects of the aldoses. So aldoses are the sugar which contain a aldehyde group as a functional group. So these are the aldoses, and these can be the some other sub classification of these aldoses, are trioaldose, tetroaldose, pentoaldose, hexoaldose. So many different kinds of the aldoses we have recorded in the nature, and therefore we can say that this any kind of a sugar or any kind of a mono sugar which contain a aldehyde as a functional group, then it is called as a aldoses. And so these aldoses or the functional groups are very important in the function of that mono sugar as well, and therefore we have to think about the aldoses as well. There is another type of a functional group we have mentioned in the which is present in the monosaccharide, and that is a ketone. If the ketone is a functional group, those kind of sugars are called as a ketones. Ketones are the sugars, ketones are the monosaccharide which contain ketone as a functional group. And so this ketone is a functional group, CO is a functional group, then it is called as a ketones. These ketones can also be subclassified based on the number of carbon atoms present in the ketones, and therefore we can classify them as that of the aldoses. So the first you can say if 3 carbon sugar contain a ketone as a functional group, those can be called as a ketotriose. Example we can take a dihydroxyacetone. Dihydroxyacetone is an example of the ketotriose. Then you can take another, if 4 carbon numbers are present in the sugar and it contains a functional group that is a ketone, then that can be called as a ketotetrose. that can be called as a ketotetrose. Ketotetrose, erythrulose is an example. If 5-carbon sugar, 5-carbon monosugar is there and contain a ketone as a functional group, 4-carbon first, so 5-carbon, then it is a pentose, and that pentose, if contain a ketone group as a ketone as a functional group, then it is a ketopentose. Ketopentose, form of a aldehyde into the ketone, or it is a isomer of the aldehyde type of a pentose sugar, that is a ribose, and that you can take an example here as a ketopentose, that is a ribulose. And when we look into the hexose sugar, that is a ketohexose. It is a 6-carbon sugar, 6-carbon monosugar containing the ketone as a functional group, and there is an example of a 6-carbon sugar, that is a fructose, is a ketone sugar. So if we are thinking in the many more carbon also, just put a keto as a prefix into the number of a carbon sugar based on that different kinds of the classification, then we can understand that these different kinds of the sugars are containing the ketone as a functional group. So finally we can take a overlook for the different kinds of the aldoges and ketoges. So, and the examples as well we will think in detail. So these are the different kinds of the sugars or monosugars or monosaccharides we can say, and those contains aldehyde or ketone as a functional group, and based on that they have different kinds of the functions as well, and therefore we have to think about the different kinds of the aldoges and the ketoges. So this is the one of the classification, but monosugars can also be characterized, or their characterization can be very much important, and another important character of this monosugar can be considered in the understanding, that is alpha and beta conformations. Alpha and beta conformations. What are the different alpha and beta conformations? When just simply, many times you will see that the sugar, monosugar is a alpha sugar, monosugar is a beta sugar. Also there is a dextro sugars and leo sugars. There another stoichiometric examples we can see, and labeling also there. D is for dextrorotatory, and D for dextrorotatory you can see, and L for leorotatory. You may have clear your concept of the dextrorotatory and leorotatory. If the sugar, if we pass a monochromatic light through the sugar, at that time if light passes or deflect its path towards the right side, then it is a dextrorotatory, and if it is light, when monochromatic light will pass through the solution of this sugar, monosugar, and if light deflects to the left side, then it is a leorotatory. Simply, when we look at the beta conformation is the conformation at the carbon number 1, or their functional group where it is present, and what is the conformational position of the functional group. So that conformational position of the functional group is very important in the functional aspects of the monosugar, and therefore we will be thinking in the whether the sugar is alpha or beta. So very simple example we take, which is the alpha and which is the beta, and when we take a example of a alpha sugar, especially of a glucose, then we can think in the different way that glucose is a hexose sugar, and it contain normally it present in the ring structure, and that ring structure contain a 6-member ring, but one of the member is a oxygen, and therefore we will put oxygen as it is. So normally there is a alpha conformation of a glucose, and then we put the hydroxy group at the lower side. Then another, we can take an example of in different way, that OH group is present, sorry, H is present, OH is present, OH, CH2OH. So this will be the very simple structure of a sugar, that is a glucose. And when the OH group is present at the lower side, simply you can say lower side, and this is called as a alpha glucose. We are talking about a D-glucose only. D-glucose or a dextrorotatory glucose is normally or naturally found in the nature. It is a naturally found glucose molecule, that is a D-glucose, and its conformation, if it is at the lower side, then you can say simply the alpha glucose. But when we take a same example and we see what is the beta conformation, then we can understand the different aspect of this mono sugar. If OH group is present in this way, CH2OH. So in this case, what happened? So OH group is changed, and therefore their beta conformation is changed, and that conformation is a now beta conformation, and therefore this sugar is called as a beta glucose. So it's a very simple understanding. If in any sugar or any type of a mono sugar, if first functional group contain a oxygen or hydroxy group at the right side, then it is a alpha glucose, and if it is present at the left side, then it is a beta glucose, or upper side it is a beta conformation, and lower side it is a alpha conformation. So this conformation also we will be thinking in the different way for many different kinds of the aspects in the composition of other kind of carbohydrates as well, and different kinds of the other aspects, especially in the functional type of aspects as well. So these are the another kind of criteria we consider for the classification of the monosaccharides. So these are the different aspects we have discussed about whether this mono sugar contain aldehyde group or ketone group. Based on that, these mono sugars are classified into the two different kinds of the group, aldoses and the ketones. If its conformation is whether beta or alpha, and as well as the dextrorotatory or levorotatory. Then these mono sugars also have a special kind of a property. They have a free OH group or a free aldehyde group for the aldehyde or ketone group for reaction, to do reaction with the different kinds of the other reagents as well. So based on the reactivity, these sugars can be classified into the two different based on reactivity. So how this sugar reacts with different kinds of the reagents. So for example, if this sugar has a reactivity of a reduction type, reducing power, it has a reducing power. If it has a reducing power, this must have the aldehyde and ketone as free groups to react. If it has a aldehyde and ketone as a free group to react with the other or to reduce the other kind of reagents at that time, these kind of sugar are called as a reducing sugar. These are the reducing sugars. Because they contain free aldehyde and free ketone group, and therefore this ketone group and aldehyde group can react with the other kind of a reagent. They reduce the different kinds of the reagent such as a Benedict's reagent. Benedict's reagent. This Benedict reagent contain a copper ions, diionic copper. It will be reduced into the copper 1, and therefore, or you can say cupric ions are reduced into the copper, and this reduction is done by the CHO, free CHO or free ketone group, and therefore these two different kinds of the group, if freely present in the monosugars or monosaccharides, then they have ability to reduce the copper ions present in the Benedict reagent, and therefore these kind of sugars are called as a reducing sugars because they have a reducing power, and therefore they are responsible for reduction of different kinds of the oxidizing agents such as cupric ions, and therefore cupric ions are converted or are reduced into the cuprous ions, and therefore you can say these kind of sugars are the reducing sugars. In simple way, we can say all monosaccharides are reducing sugars. Remember, all different kinds of the monosaccharides, maybe it is a glucose, fructose, glucose, fructose, galactose, or any other kind of monosaccharides. All those monosaccharides are reducing type because they contain their free CHO or free aldehyde group, CHO or aldehyde group, as well as ketone as a free group, and therefore they have ability to reduce the any kind of a reagent such as a Benedict's reagent, and therefore those are called as a reducing sugar. So all different kinds of the monosugars are reducing sugars, you can say. But when we think of the other kind of criteria for the reducing sugar, then other further classification, that is a disaccharide, trisaccharides, or polysaccharides, those are may or may not be a reducing sugar, but all different kinds of the monosaccharides are reducing type, reducing sugars, or you can say these are the reducing. Just to have examples such as glucose, fructose, galactose are the sugars which can be considered as a reducing sugars, and these are all monosaccharides, and so these monosaccharides are the reducing sugars. So these kind of criteria or properties, chemical property of these monosugars are also need to be considered, and so these kind of reagents such as the Benedict reagents are used, or this property of having a free or free sugar, free functional groups such as aldehyde and the ketone group, those are used in the quantification of the monosaccharides. So how much glucose is present in the blood, that can be assessed based on the number of functional groups, such as when we talk about the glucose, this CHO group, whether it is present and how much is the amount of glucose is present based on the is determined based on the how much reagent is reduced, and that color formation is finally converted into the amount of glucose. And so this kind of a reaction is utilized in the determination of the glucose level in the blood, and therefore we can say that reacting properties or chemical property of these monosugars are utilized in their quantification as well, so determining the amount of sugar in the blood. And so these reducing sugars are all different kinds of the monosaccharides. Again, you can think other kind of reducing sugars when we talk about the other kind of different types of mono or other kind of carbohydrates. So these different kinds of the monosaccharides, we have got different ideas about the monosaccharide. When we think about the specific type of monosaccharides, especially very common monosaccharides, say glucose, galactose and fructose, then we can understand these are the different types. So the first is the glucose. Glucose is a aldohexose. Remember, aldohexose. It says that it contains a functional group aldehyde as a functional group, and hexose says it is a 6 carbon sugar. So it's a very simple, that is aldohexose contain aldehyde and 6 carbon number. When you look at the different aspects of this or different properties of this glucose, glucose is normally present in the blood. Normally present in blood, and so it is also called as a blood sugar. Blood sugar. So how much is the quantity of a sugar, this glucose present in the blood? Normally it is about 90 mg per 100 ml of a blood. Its concentration or its level is about 90 mg per 100 ml of a blood. So this is the normal blood sugar. If it increases, it may have a different kinds of the physiological changes, or if it decreases, it also have many different kinds of the adverse effect on the body. So why and what is the importance of this monosugar? It is very simple. This glucose is a very simple monosugar, and therefore it can pass through. It can pass through membrane. Pass through membrane. It can pass through the membrane of a cell, and therefore it can freely pass through the plasma membrane, and therefore it can be utilized by the cell, it can be given out by the cell, and therefore it has a great importance. When we eat a glucose, at that time it is freely absorbed in the intestine as well, and therefore this sugar has a great importance in the physiology. This is the common sugar which releases the energy. It is used for the release of energy. Release of energy. Especially it releases the energy in the form of ATP, adenosine triphosphate, and therefore it provides energy for all different kinds of the bodily processes or metabolic activities, and therefore this glucose is at the center of the metabolic activity or energy metabolism, we can say. So central sugar at the energy metabolism is the glucose, and it is a monosaccharide, or you can say it is a aldohexose. So this is the important sugar, important monosugar, we can say, or monosaccharide. Second type of monosaccharide, that is galactose. Galactose. It is an isomer of a glucose, but it is a beta isomer of a glucose, we can say. This beta galactose, especially beta conformation of a galactose, it has the same number of carbon as that of the glucose. It has a aldehyde. It is also aldohexose. Aldohexose. It is also aldohexose because it also contains a aldehyde as a functional group and 6 number of a carbon in their structure. It has a no much important role in the physiology of a living organism, but it has a very important aspect in the formation of a one important disaccharide, that is it combines with, especially it combines with glucose. This galactose combines with the glucose and leads to formation of a lactose, and therefore this lactose is a milk sugar, and therefore this is very important in this case. Especially it is lactose is present in the milk, and this lactose is formed of glucose and the galactose, and therefore only for the formation of this lactose, this galactose has a importance, and therefore this is very important in the nourishing the baby or nutrition of the baby or nourishment of the baby. So this galactose is important in only one case, but other kind of aspects, that is energy metabolism, because it has a different kinds of the conformation, and therefore this conformation leads to its difficulty in the utilization in the different kinds of the physiological processes, and so it is not used anywhere else. So this is the galactose. Third type of monosugar or monosaccharide we do have, and that is a fructose. This fructose is also a ketoaldose, as we have said earlier. Ketohexose. It is a ketone isomer of this glucose only, we can say. It is a functional group which is completely different in the fructose, as we have seen that glucose contains aldehyde as a functional group. This fructose contains a ketone as a functional group. So it is a ketone as a functional group, and as hexose represents a 6 carbon sugar. So this fructose specially called as a fruit sugar. It is a fruit sugar, and because it present in the fruits. So this monosaccharide which is present in the fruit is called as a fruit sugar, and it is a fructose. This fructose combines with the glucose, and forms a sucrose. And therefore this fruit sugar is very important in the formation of another kind of disaccharide, another kind of carbohydrate, and that is a sucrose, and therefore it is also important. It also plays a very important role in the physiological chemistry or biochemical different reactions as well, and therefore fructose is very in terms of importance, it is next to the glucose, and so fructose is also important sugar. So this is also a monosugar. So these are the different kinds of the monosaccharides. So in the monosaccharides we have discussed type of monosaccharide based on the number of carbon, based on the functional group, based on the conformation alpha or beta, based on their stoichiometric properties, the dextrorotatory or levorotatory, and then we have also discussed about some common examples of the monosaccharides. So this is about the monosaccharide. Then we will turn towards the another important point, and that important point is the second type of carbohydrate, and those second type of carbohydrates are nothing but the disaccharides. Second type of disaccharides. As monosaccharides are the simplest sugar and which acts as a unit for the different ends of the other carbohydrates. But disaccharides are the sugars which are formed from the monosaccharide. When these disaccharides are the sugars, these are the sugars formed of condensation of two monosaccharides. Remember, two different monosaccharides or two similar monosaccharides condensed together and forms a disaccharide. Therefore, this 2 is represented by the di 2. Therefore, these are called as a disaccharides. So, this condensation reaction occurs by removing the hydrogen atom. Hydrate group or the water molecule, we can say. Hydrogen and oxygen molecules are removed from the these two different ends of the sugar combination, and therefore that process is called as a condensation. So condensation is the formation of a bond between two different ends of the molecules by removal of a water molecule, and that process of condensation is used for the formation of a disaccharide. So first we will see how this condensation happens or occurs in terms of some other kind of two monosaccharides. So when we think of the simple and very common sugar, that is a hexose, that is a glucose, then two glucose molecules, alpha glucose, we can say glucose plus another alpha glucose comes together and forms a disaccharide, and that disaccharide can be a maltose. Remember, maltose. And how does it happen? If we represent with the general formula at that time, C6H12O6 plus C6H12O6, it will leads to the condensation. Remember, condensation, and in the condensation there is a removal of a water molecule, and therefore it will form other kind of important sugar, that is a disaccharide. We can represent this disaccharide by C12AH22O11. So like this disaccharide can be represented, or disaccharide can be represented in other way as well. Other way, you can see what is the other way. C6 or Cn, general formula we can put Cn H2n minus 2 On minus 1, and it is a 2. So N is 2, we can see N is 2. So that N is always 2 in the disaccharide. Or you can represent this in many different other way as well, based on the what kind of a sugar you are using. So it can be a general formula, it can be specific for the hexose disaccharide. Hexose disaccharide are the disaccharide which are formed of the hexose sugar, and therefore this becomes a different kind of a formula. So when we look into the structural formula at that time of the glucose only, we can say this glucose molecule represented as CH2OH OH, sorry, H, OH, OH, H, H. Okay, this combines with other kind of a same mono sugar, that is a glucose, CH2OH. When these two different kinds of the groups combines together, same alpha glucose comes together, they form a different kinds of structures, and that structure is nothing but the disaccharide. So OH, OH, CH2OH, OH, CH2OH. So what happens? These two different kinds of the group, these two different kinds of the groups are responsible for making a condensation reaction, or these two groups, remember this group and this OH group takes part in the condensation reaction. So what happens? First we will see how this group, one of the group, or they can attack on each other. They attack on each other, and therefore they leads to the condensation. Then what will be happen? Removal of the one OH group or one oxygen and two hydrogen atoms from this, and therefore it leads to the removal of a H2O. So say this OH group plus this hydrogen group, OH plus H, is removed, and therefore this, you can see there is a formation of a bond which contain only oxygen. So therefore removal of a water molecule is the condensation reaction and joining of two different kinds of the monosaccharides, and these monosaccharides are now combined together and form a disaccharide. So it's a alpha glucose, another plus alpha glucose, say removal of water, and then it is a disaccharide maltose. In this, glucose is a monosaccharide. It is also monosaccharide. Both the glucose are monosaccharide. These two monosaccharides comes together and form a disaccharide. So maltose is a disaccharide. This both the monosaccharides or two monosaccharides joined together with a special kind of a bond, and this bond is called as a glycosidic linkage. So this glycosidic linkage is very important for bringing together two different kinds of the monosaccharide, and therefore these monosaccharides are joined together with the glycosidic linkage. So we can say that this is the disaccharide. and disaccharide contains a glycosidic linkage, one glycosidic linkage. In a disaccharide, in single disaccharide, there is a single glycosidic linkage, and this linkage can be termed in the different way. So, what is the other aspect of this formation of a glycosidic linkage? That is, this glycosidic linkage contains or involves different kinds of the carbon atoms. So, it involves two different kinds of carbon atoms. So, when we give a numbering to the carbon atom, then it is a first carbon atom, second, like this. It is the first carbon atom, second carbon atom, third, fourth, fifth and sixth, like this. Same numbering you can apply for the other kind of glucose as well. So, likewise, one first or OH group of a first carbon atom of a first sugar, mono sugar, especially glucose. First carbon atom, OH of a first carbon atom of a first glucose reacts with a OH group of the fourth carbon atom of second sugar, and therefore these two numbers are involved, and therefore this linkage can be named based on the such kind of the involvement of the carbon atoms and their OH group. So this number of carbon can be given to the glycosidic linkage. So this kind of a glycosidic linkage is alpha first and fourth. Alpha first carbon is attached to the fourth carbon. Both are in the alpha conformation. Remember, both are in the alpha conformation. If conformation of this group is different, that is a beta, then it may have been a beta 1,4 linkage. But it is a alpha 1,4 linkage. And this alpha 1,4 linkage is a type of a glycosidic linkage. Remember. So, in the maltose, there is a alpha 1,4 linkage. And therefore, such kind of the examples we can take. And this process, complete process, is nothing but the condensation process by removal of the water molecule. And so, single monosaccharide and other monosaccharide, that is a two different kinds of monosaccharide, comes together and forms a large structure of a carbohydrate, and that is a disaccharide. So, this disaccharide is converted into the, monosaccharides are converted into the disaccharide, and therefore these disaccharides are larger in structure, therefore they are unable to pass through the plasma membrane. They are unable to pass through the plasma membrane. Another aspect we have to discuss here, that some of the disaccharides contains different kinds of the functional group, whether may be freely present or may not be freely present. So, when we look at this mono sugar, at that time, this functional group is a free, or this specially, we can think in the different way, or I will draw the separate structure of a monosaccharide. Say it is a glucose. CH2OH. This functional group, this complete functional group is free, and therefore it is a reducing. In some of the disaccharides, this OH group is also free. One of the OH group at the next or right side or in the joining side, you can see. Here, this functional group is free. This functional group is a free, especially in the maltose, and therefore maltose is a reducing sugar, and therefore this sugar, this disaccharide, or you can say this is a maltose, and this maltose is a reducing type of a sugar. It is a reducing sugar. Why it is reducing? Because it contains a free aldehyde group to react with a reagent, and therefore it is a reducing sugar. This disaccharide reducing sugar. Other kind of reducing sugars you can take examples that lactose is also reducing sugar because it contains a galactose and the glucose, and free aldehyde group of a glucose is ready to react with, or it is free to react with a reagent, and therefore galactose is also lactose. Another you can say maltose formed of a glucose linked to glucose, and therefore there is free OH group. There is a free CHO group, free CHO group, and therefore it is also reducing sugar. When we look at the other kind of sugar, lactose is composed of the galactose linked to glucose, and this glucose have a free CHO group, and therefore it is also reducing. Maltose also reducing and lactose also reducing sugar. So these are the two different kinds of the disaccharide which are reducing sugars. But when we look at the other kind of groups or other kind of sugars such as sucrose, in a sucrose, glucose is linked to fructose, and it contain no free C or ketone, C double bond O or ketone group, no free ketone to react with the reagent, and therefore there is a no reducing power in this sugar, and therefore it is called as a non-reducing sugar. It is a non-reducing sugar. This sucrose is a non-reducing sugar. So lactose and the maltose both are the reducing sugars, and sucrose is not a reducing sugar, and therefore we can understand that this disaccharide can also be of two different kind, that is a reducing or non-reducing. This sucrose is a non-reducing, but maltose and the lactose are the reducing sugars. But other kind of disaccharides are hardly reducing type. So most of the disaccharides are the non-reducing type. Polysaccharides are also non-reducing type, and therefore we can classify these different kinds of the disaccharides into different reducing and the non-reducing groups as well. This disaccharides contain two different kinds of the monosaccharides or similar type of monosaccharides as well. So these monosaccharides forms two monosaccharides forms a disaccharide, and therefore it becomes a larger structure, or their size is comparatively larger or double than the monosaccharide, and therefore they cannot be freely, when this disaccharide is present within the cell, it cannot pass out of the cell, or it cannot enter into the cell if it is present outside the cell, and therefore they can't pass through the plasma membrane. So how these disaccharides are naturally found in the different kinds of the plants, or it is a natural sugars, or all different kinds of the carbohydrates. Monosaccharides can't be found naturally as a free, but disaccharides are naturally found sugars, and therefore these naturally found sugars are available for the eating for the different kinds of the animals. And so if these different kinds of the animals eat the disaccharide, then how does they absorb the sugar into the cell for their utilization in the energy synthesis or formation of different kinds of the energy molecules, or providing the energy for the metabolic reactions. How does it happen? So this can be done by the hydrolysis of this disaccharide. So hydrolysis is the another process by utilization of a monosaccharides. So disaccharide can be hydrolyzed. In the hydrolysis, water molecule is utilized. Water molecule is used and form a 2 monosaccharides. And now these 2 monosaccharides are absorbed in the body. So during the digestion, there are many different kinds of the enzymes which do hydrolysis of a disaccharides by using or by adding the water molecule. They break down the glycoside linkage between the 2 different kinds of the monosaccharide and they make a free monosaccharides. So disaccharide is converted into the 2 monosaccharide by addition of a water molecule, and that is a hydrolysis. Hydrolysis is the process of a breakdown of a molecule by use of a water molecule. So these are the different aspects of the disaccharide. So these disaccharides are commonly found and utilized. And so we can finally come to the examples of this. Already I have discussed different kinds of the examples of this disaccharide. Common examples: sucrose. Sucrose is a cane sugar and normally present in the different kinds of the plants or plant parts. Sucrose is formed of a glucose and fructose. Glucose and fructose combines together forms a sucrose. Second example, maltose, we have discussed in detail. It is formed of a glucose combines with a glucose. Third common example of this disaccharide is a lactose. Lactose is formed by galactose and glucose. So these are the 3 different kinds of the common examples of the disaccharide. So these are the disaccharides. So disaccharides are utilized in the many different kinds of the processes. Next type of important classification we have to discuss. Next to the disaccharide, there is another example or another type we are taking into the consideration. Another important example of our classification or another important class of carbohydrate we are going to discuss, because it is not mentioned in the textbook, but we have to discuss this kind of a class, because if we add 2 different kinds of the monosaccharide together, that becomes a disaccharide. If we add 3, then what is that? If we add 4, then what is that? If we add 5, 6, 7, 8, 9, 10, then what? Because when we talk about the polysaccharide, it contains a many number of monosaccharides in their structure. But when we add a 3, then what is that? So another important class is derived, that is a oligosaccharides. Oligosaccharides. These oligosaccharides are the carbohydrates which formed of 3 to 9 monosaccharides are formed of combination or condensation of formed of by condensation of condensation of 3 to 9 monosaccharides. It contained about 3 to 9 different kinds of the monosaccharides. 3 means if it contains a 3 monosaccharide, if 3 monosaccharides, then it is a trisaccharide. Trisaccharide. If oligosaccharide contain 3 number of monosaccharides, then it is a trisaccharide. If 4 monosugars, 3 monosaccharides are present, 4 monosaccharides are present, then it is a tetrasaccharide. Tetrasaccharide. So 3 monosaccharides, trisaccharide. 4 monosaccharides, tetrasaccharide. So based on that, if 5 different monosaccharides comes together or condense together, it will form a pentasaccharide. Then 6 hexasaccharide. Like this, many number up to the 9th. If the number of monosaccharide combined together between 3 to 9, those are called as a oligosaccharide. Number of monosaccharides present in the oligosaccharide ranges from 3 to 9, and therefore it is completely different from the disaccharide, because in the disaccharide only 2 are there. In trisaccharide 3 monosaccharides are there. In tetrasaccharide 4 monosaccharides are there. Pentasaccharide 5 monosaccharides are there. In hexasaccharide 6 will be there. Like this, many different classification you can do, and these are the oligosaccharides. So oligosaccharides has completely different types of properties as compared to the disaccharide. But when we think of the oligosaccharide, what are the different examples? We will be thinking the naturally occurring oligosaccharide. Naturally occurring oligosaccharides are only of 2 different types. One is a trisaccharide and another is a tetrasaccharide. Those are the naturally. But other kind of oligosaccharides are synthetic one, and therefore we need not to discuss about those different kinds of the oligosaccharides. But what is the example of a trisaccharide which is naturally occurring? Naturally occurring trisaccharides, its number of monosaccharide, I will write here 3 to 9 monosugars are condensed together and therefore forms a oligosaccharide. Condensation of. So first important is the trisaccharide. It contains 3 monosugars. And important example is raffinose. Raffinose is a trisaccharide, and this is composed of 3 molecules or 3 monosaccharides. Raffinose is formed of 3 monosaccharides, and in this 3 monosaccharide, one is a galactose plus glucose plus fructose. It is mostly naturally occurs in occurs in vegetables or beans. You can see this raffinose is a trisaccharide. Raffinose is a trisaccharide containing 3 monosaccharide. Especially in the raffinose there is a galactose, glucose and fructose, and therefore this different kinds of the 3 monosaccharide comes together and forms a raffinose. We can see it is hard to cut or break down in the human being, but there are many different kinds of the other acids which can hydrolyze this trisaccharide into the 2 different kinds of the group. One is a galactose. Galactose is removed by the galactase enzyme and forms a galactose and the another, that is a sucrose. So sucrose can be synthesized from this raffinose, and so raffinose is utilized for the different kinds of the energy formation or energy energetic activities as well, or energy metabolism as well. So glucose and fructose is used in the energy metabolism. So naturally, where it found, this raffinose is found in the different kinds of the vegetable as well as different kinds of the beans, such as soya. But soya bean also contain other kind of sugar, and that is especially 4 monosaccharide or 4 monosugars are present in that, and so we will be discussing soya bean in the next. So beans contains raffinose, different vegetable contains raffinose. Other kind of sugar or oligosaccharide we can see, that is a tetrasaccharide, naturally occurring oligosaccharides we are talking about. Tetrasaccharide. This tetrasaccharide is formed of 4 monosaccharides. So tetrasaccharide contain 4 monosaccharide, and these 4 monosaccharide, you can say there are different kinds of the example. One example we can take, that is a stacchylose. This stacchylose, you can see, is formed of 4 monosugars. 4 monosugars are present in this. In this case, what are the different kinds of the 4 monosugars? 2 galactose are present. 2 galactose are present, plus 2 molecules of galactose present, plus 1 molecule of glucose, plus 1 molecule of fructose is present, and therefore 2 plus 1 plus 1 becomes 4, and therefore 4 monosaccharide comes together and forms a stacchylose. This stacchylose is commonly found in the soya bean. Soya bean contains large amount of the stacchylose. Also it contains a raffinose as well, but stacchylose is another type of oligosaccharide present in the soya bean. So these trisaccharide and the tetrasaccharides are the naturally occurring sugars, or we can say those are the oligosaccharides, because it contains 3-4 number of monosaccharides. So these kind of sugars or these kind of carbohydrates are the oligosaccharides. So oligosaccharides, you can simply understand that these are formed of 3 to 9 monosaccharide condensation, formed by the condensation of 3 to 9 monosaccharide. So these are the oligosaccharides. And the next important class of this carbohydrate is the polysaccharides. Another important class of this is the polysaccharides. These polysaccharides are the carbohydrates which are formed of the many, poly means many monosaccharides are present. Monosaccharides are present. There are many monosaccharides are present. Many means 10 to many, 10 to 100 or 1000, or you can say more than 10 monosaccharides are present. More than 10 monosaccharides are present, because one are the monosaccharides. Single monosaccharide units are the monosaccharides. Those are simple sugars. If 2 monosaccharides comes together, that is a disaccharide. If 3 oligosaccharide, that is a tri. 3 to 9 we are considering, or we are including them into the special group, that is the oligosaccharide. But when more than 10 or more than 10 monosugars comes together, it forms a polysaccharide. These polysaccharide can be formed of or formed by condensation or series of condensation. Series of condensation reactions. Series of condensations. Or you can say many or serial addition of the or formation of a bonds between the monosaccharide. Suppose one monosaccharide condensed together, condensed with the other, it becomes a disaccharide. Disaccharide condensed with one more monosaccharide becomes trisaccharide. Trisaccharide condensed with one more monosaccharide, tetrasaccharide. Tetrasaccharide combines with another monosaccharide unit, penta. Likewise, hexasaccharide, heptasaccharide, octa, nona, deca, and polysaccharide. Likewise, you can simply say that these monosaccharides comes together in the large number, and therefore it is called as a polysaccharide. So when we look at the different kinds of the aspects of this polysaccharide, these polysaccharides are formed of the condensation. So similarly, here also the bond between the two different kinds of the monosaccharide in the polysaccharide is also a glycosidic linkage. Glycosidic linkage. These glycosidic linkages are formed between the two sequential or two side or two adjacent molecules of the monosaccharide. So these glycosidic linkages are present in large number in the polysaccharide. So this can be considered as a condensation or series of a condensation of a monosaccharides leads to the formation of a polysaccharides. So these polysaccharides are containing a different kinds of the monosaccharides or same type of monosaccharides as well, and therefore what kind of a nature of a linking or a linkage, there may be the linear polysaccharide or branched polysaccharide. The linear polysaccharides, in this case the monosaccharides are joined in the linear fashion or one next to the other. Likewise, so you can see n number can be there. So likewise, repetition may be of a single, repetition may be of a two. Likewise, many different kinds of the combination may be present in the linear fashion of the polysaccharide or polymerization. Polymerization can be a linear or polymerization can be a branched. When there is a linear type of a linkage, at that time properties can be of a different types. In case of the branched polymers, their properties are also completely different than the linear one, and therefore based on the linear arrangement or a linear polymerization or the branched polymerization, their properties are also completely different. So we will be discussing what are the different types of linear polymerization and what is the different types of branched polymerization as well. So when we look at the linear polymerization, at that time the different kinds of the monosaccharide molecules are joined together, and that kind of a group, if it is formed of a glucose only, then it can be considered as a amylose. So one of the example of the linear polymerization in the polysaccharide is the amylose. So amylose is a polymer of glucose. It is a polymer of a glucose molecule. So what happens? We can draw a structure of this amylose when it is a linear fashion. CH2OH. Likewise, it will combine with the other kind of a glucose molecule, and therefore it becomes a you can see disaccharide. Now CH2OH, and in next there can be the O. One more we will draw. Like this. So OH, H, OH, OH, EH, EH. Like this. OH, OH group, H, H. Here you can see H, OH, H, OS, H, OH, H, H. Hydrogen will be there. Hydrogen will be there. Like this. So what happens? This unit is repeated many times. This unit, middle unit, can be repeated n times. This can be a n number. So what happens? These two molecules of a glucose joined together, and this branch is, or this linkage is the 1-alpha-1,4 linkage, and therefore amylose is formed of the many linear arrangement of the glucose molecule, and therefore each glucose molecule joined together with the other molecule, especially alpha-1,4 type of a linkage. So alpha-1,4 type of a linkage is present in the amylose, and so this structure is completely consequently repeated. That is a n number of a middle glucose residues. Now we can say it is residues. So n number of a glucose residues are present. So like this, you can see formation of a linear polymer. When we put this amylose into the water or into the hot water, it forms a colloidal solution. In hot water forms colloidal solution. Because this linear polymer becomes coiled in the hot water, and therefore that solution becomes a colloid. Otherwise these are insoluble in the water, but it forms a colloidal solution when we put it into the hot water, and therefore you can see this linear arrangement can be of a different type as well. Maybe a coiled one, like this linear will be there, coiled will be there, like this. So this only one transformation will occurs in presence of the heat, but if we heat this kind of amylose, it will not break into the smaller units, but if we do hydrolysis of this, it will result into the formation of the monomeric unit. So remember, linear arrangement in the amylose is with the bond that is called as a alpha-1,4 linkage. When we look at the other type of a polymerization, this another kind of a polymerization can be branched polymerization. An example of a branched polymerization is a amylopectin. Amylopectin. In amylopectin, what happens? There is a branch, but when there is a branching, the branching occurs in between the first carbon and that of the sixth carbon. So, because of that only we can think in the different way. So, when we think in different way, at that time we can understand that there is a formation of a branch, and this branch can be part of a many different kinds of the groups. There can be the different kinds of the groups. So I will draw one glucose molecule here. You can see here CH2OH. But now what happens? CH2OH. Likewise. So here sixth carbon of a one glucose molecule binds with the first carbon of a other. Therefore, this hydrogen atom and oxygen atoms, this condensation. So this linkage is a alpha 1,6 linkage, and this is the branching point. This is the branching point, and this branching point leads to the formation of another kind of a polysaccharide, another type of a polysaccharide, that is a amylopectin. And here you can see these units can be also repeated n times. So there is a formation of a bond like this. There can be the first carbon atom OH can link to the other, and therefore this linkage, this branched point is nothing but the 1 alpha 1,6 linkage, and therefore this branching can vary from type of a polysaccharide, and this is the amylopectin. And amylopectin, because of its branching point, it is insoluble in the water. So amylopectin is completely insoluble in the water, and therefore we can think these polysaccharides can be branched or linear. Linear polysaccharides, especially examples you can see, if it is formed of a glucose, then it is a amylose, and branched polysaccharide, if it is formed of a glucose, then it is a amylopectin. So there are different kinds of the polysaccharide based on the what type of monomeric units they contain. So polysaccharides can be divided into the two different groups. One is the homopolysaccharide. One is the homopolysaccharides, and other is the heteropolysaccharide. Heteropolysaccharides. In the homopolysaccharides, monomeric unit or single monomeric unit is present. Single monomeric unit is there. Single monomeric unit. Remember, single monomeric unit means only one type of monosaccharide is present. Monosaccharide present in its polymer. Remember, single monomeric unit means only one type of a monosaccharide is present in the homopolysaccharide. Maybe if we say all different kinds of the monomeric units are only glucose, if it is a only glucose, or only galactose may be there, only fructose may be there, like this, only one type of a monomeric unit is present in the homopolysaccharide. In other kind of a polysaccharide, that is heteropolysaccharide, different types of monomeric units are present. Or different type of monosaccharides. Are present. Remember, if only one type or single monomeric unit is present, then it is a homopolysaccharide. If different types of monomeric units are present or different types of monosaccharides are present, then it is a heteropolysaccharide. There are different kinds of the examples of this polysaccharides. If it is a homopolysaccharide, there are three different common examples we can see. That is, a starch is also a homopolysaccharide. Then we can think of the glycogen, another, and third, cellulose. In the starch, monomeric unit is a glucose. Glucose is monomeric unit. In the glycogen also, monomeric unit is a glucose. In cellulose as well, monomeric unit is a glucose. So only glucose is a monomeric unit of a starch, glycogen and the cellulose, but they are of different type that we will discuss. When we think of the heteropolysaccharide, there are many different kinds of the examples. Say that is a heparin, is a common example of this heteropolysaccharide you can discuss. Or blood groups, or the carbohydrates present on the RBCs or blood cells, those are also heteropolysaccharide. Heparan sulfate can be there. Heparan sulfate. That is a sulfated monosaccharide unit is present in the heteropolysaccharide. So heteropolysaccharide are present in the different types of cell in the different forms, and therefore we cannot define what are the different. There are large number of the different monosaccharide units are present. Maybe they contain fructose, glucose, galactose, and many other sulfated sugars are there, amino monosaccharides are there. So modification of the monosaccharides is there in the heteropolysaccharide. Therefore it is not need to discuss in detail, that is what different kinds of the monomeric units are present in the different heteropolysaccharide, but definitely we will discuss what are the different kinds of the monosaccharide units are present in the starch, and what are the starch, glycogen and glucose or homopolysaccharide, and what are the different properties of those polysaccharides, homopolysaccharides especially, and how does they have a speciality in their structure, that we will discuss first. So first important homopolysaccharide is a starch. This starch is formed of two different kinds of the polysaccharide, one linear and another branching. Combination of both linear and branching. In the linear, it is formed of glucose, therefore it is a amylose, and in the branched, that is a amylopectin. Remember, amylopectin. It is formed. The branching is the same. Here only alpha 1,4 linkages are present in the amylose, and in the branched both are present, especially in the branching point, especially when we talk about the amylopectin, then it contains both alpha 1 4 linkages in the straight line and alpha 1 6 linkages as well. So both are present in the amylopectin. So the branching or glycoside linkages in the starch can be alpha 1 4 and the alpha 1 6 linkages. And therefore we can also think that the starch is a common storage food material. Storage food in plant. Starch is the storage food material in the plant. So it is a storage food of a plant, and therefore it naturally occurs in the different kinds of the plants. So we can see natural source of this starch can be the potato. Potato is a very rich source of a starch, and therefore if you want a starch, that you can put some potato into the water, and starch will get dispersed in the insoluble form, and therefore it becomes a turbid. So this starch can be extracted from the potato, and many other kind of such kind of the modification of the plant parts will have or may contain store this starch as a storage food, and therefore we can collect it from the such kind of the parts. So vegetables also contain a starch, fruits also contain a starch, and many other different kinds of the parts, plant parts, stem, root also contain a starch. Everywhere, especially in the stems, stem stores the food material in the form of a starch. Leaves also stores the food material in the form of starch, and therefore starch is the storage food in the plant, and therefore it contains different kinds of the. So another important aspect of the starch here, it has a branching, less number of a branching. It contains a less number of branching. Remember, its branching frequency is one branch at every 20 to 30 glucose molecule. Remember. So if we see glucose molecules are there and branch is there, so both branching point is there and linear glucose molecules are also arranged. When we look at this, 1 2 3 4 5 6 7 8 9 10 11 12 13 14 15 16 17 18 19 20 21 22. So every 20, say here is the branch, and another we can see here is the branch. Every 22 30, maybe it more, branching point is present at every 20 to 30 glucose molecule. So if 20 glucose molecules are present in between the, or you can say simply say 20 to 30 glucose molecules are present in between the two different branches of the this amylopectin or in the starch. In starch, if there is a branch present, and this branch is present at every 20 to 30 glucose molecule, and therefore it has some difference than the other kind of a homopolysaccharide found in the animal. So we can simply say the other kind of a or second type of a homopolysaccharide, that is a glycogen. Remember, glycogen contains a glucose as a monomeric unit. Glucose is the monomeric unit in the glycogen. This glycogen also contains both linear and branched type of a homopolysaccharide. In the linear homopolysaccharide, there is amylose, and amylose contain alpha 1,4 linkage in the glycogen as well. Branched type of a homopolysaccharide is present in the glycogen as well, and therefore this is the amylopectin. So same names are same as that of the starch. And amylopectin also contain alpha 1,4 and alpha 1,6 linkages. Therefore, this alpha 1,4 and alpha 1,6 linkages are differently present in the glycogen. This only one difference is there that we are going to change. So this glycogen is a storage put in the animal. Remember, this is the only difference. First difference we can say, it is a storage food in the animal. So where in the animal body this glycogen will be found in mostly two different organs. It is found in the liver and muscle. In liver and muscle this glycogen is present, and glycogen is stored in the two different kinds of the organ. One is the liver and another is the muscle. But here we can see the another difference in the glycogen. It contains more number of branches are present. More number of branching are present. One branch at every, remember, every 10 to 14 glucose molecule. So if we have, in case of a starch, we have branch at every 20 glucose molecule, but here it can be some more, 1 2 3 4 5 6 7 8 9 10. It may be at a 10. So this branching is very different. So therefore this glycogen is a somewhat another structure which is a densely branched, So large number of branches are present as compared to the starch in the glycogen. Starch contain a less branching and glycogen contain a more branching. That is the only difference between the glycogen and the starch. And starch is the storage food material in the plant, and glycogen is the storage food material in the animal. That is a simple two different kinds of the differences you will find in the these two different kinds of the homopolysaccharide, that is a glycogen and the starch. Third type of a homopolysaccharide, that is a cellulose. Cellulose is also a homopolysaccharide of a glucose. Cellulose. In the cellulose, what we can see, monomeric unit is glucose itself. But this glucose is not found in the alpha conformation. Instead, it is a beta glucose. Glucose is present in the beta conformation, and therefore cellulose is completely different than the starch and the glycogen. Here there is no such kind of the branching found as that of the amylopectin in the cellulose. But here two glucose molecules are joined, beta glucose plus beta glucose. Likewise, many number of the beta glucose, you can see n number of a beta glucose are present there. But these two different kinds of the glucose molecules joined together with a link or glycosidic linkage, and this glycosidic linkage is called as a beta 1,4 linkage. Here also first carbon hydroxy group or hydroxy group present on the first carbon is joined to the hydroxy group of the second glucose present at the fourth carbon, and therefore this linkage becomes a beta 1,4 linkage. And therefore you can simply say the cellulose is very different than the starch and the glycogen. Therefore in this case as well, the beta 1,4 linkage is very important, and this beta 1,4 linkage can't be broken by the enzymes present in the animal body. So this enzyme present in the animal body is a alpha amylase. and this alpha amylase can't break the beta 1,4 linkage, and therefore we can't digest the cellulose. But animals can't digest the cellulose. But in case of some ruminants, they have different kinds of the bacteria which secretes a beta amylase and therefore helps in the digestion of the cellulose. Another important aspect, this cellulose is a structural carbohydrate or structural homopolysaccharide. This structural homopolysaccharide forms a structure, or it is present in the cell wall of a plant, especially of a plant, present in the cell wall of a plant. Therefore, cell wall of a plant is composed of the cellulose homopolysaccharide. This homopolysaccharide is compactly arranged. Their linear polymers are arranged in the very compact manner, and therefore it gives its a hard structure. Therefore, cellulose has a hard structure or strong structure because of their linear arrangement of their polymers. So, cell wall is very hard or very sturdy, very strong, you can say, and therefore this cellulose forms a structural component of a cell, that is a cell. So, in many different kinds of the aspects, we can think that homopolysaccharides are present in the different kinds of the cell, and therefore it is responsible to form different parts of the cell as well as different parts of the animal. So, first two homopolysaccharide, that is starch and the glycogen, those are the responsible for, or those are involved in the storage of a food material. And third type of a homopolysaccharide, which is involved in the structural component formation in the plant, that is a cell wall. Okay. So, another important homopolysaccharide we can say, that is which is we have to discuss, that is a chitin. Chitin is another important homopolysaccharide, and this homopolysaccharide is formed of the especially alpha N-acetyl glucosamine. Remember, acetyl glucosamine, N-acetyl glucosamine. Remember, so N-acetyl glucosamine is a amino sugar. It is a modification of a glucose. Glucose is attached with the amino group, and acetyl group is attached to the amino group at the end, and therefore N-acetyl glucosamine. So, this N-acetyl glucosamine comes together in the linear fashion and forms a chitin, and this chitin is therefore is a very hard structure, and this is also a structural homopolysaccharide, and it forms a hard cover present on the body of a insect, crustaceans such as a crab and cockroaches. The body of a cockroaches is covered with the exoskeleton. It is a structural component. You can say structural component of exoskeleton of many animals, such as insects and crab. You can see this is the component of a exoskeleton of a insect and the crab. Therefore, chitin forms a hard cover around their body, that is called as a exoskeleton, and it gives a protection. So, these are the different kinds of the aspects of a homopolysaccharide. So, we have already discussed many different kinds of the carbohydrates, their types of carbohydrates. Then, what is the function of a carbohydrate? That we will see. So, this carbohydrate do have involved in the many different kinds of the functional aspects of the living organism. Especially, we can say it is involved in the energy metabolism. Energy metabolism. It is responsible for the or involved in the energy metabolism. it provides energy. You can say provides energy, especially in the form of ATP. It also provides energy to lactating baby. So it provides energy for the lactating baby, that is the lactose present in the milk provides energy for the baby when it sucks the milk. So it is involved in the energy metabolism. It is also involved in the structural component formation. It forms a structural or forms a structures, forms different structures. There are different kinds of the structures you can see. It forms a cell wall in plant. It forms an exoskeleton. It is also involved in the recognition of a cell. And therefore they forms a receptor. Receptors. They are involved in the formation of a receptors as well. So glycoreceptor, it combines with other kind of molecules, biomolecules, and therefore it forms a complicated biomolecules, and those are involved in the recognition of a cell or cell to cell contact is because of this structural forms of a carbohydrate. So it forms a different kinds of structures. It is involved in the, or it is very important in the storage of a food material. Or it acts a reserve of the food material. It is acting as a food reserves, such as a starch and glycogen, which acts as a reserve for the food material, and therefore it is stored in the different parts of the body of a living organism. Therefore these carbohydrates are involved in the mostly three different kinds of the function: one energy metabolism function, another formation of a structure, and storage of the food material in the body of living organism. Therefore these carbohydrates are very important for the living organism. So in last we can conclude that these carbohydrates are of a different type: monosaccharide, disaccharide, oligosaccharide, polysaccharides, and involved in the different kinds of function. So today we have discussed all the different kinds of aspects of a carbohydrate. Thank you very much.